Well, thanks for the introduction, Jason, and thanks for organizing the conference. It's yeah, I don't know. I think COVID can be kind of socially isolating, so it's really nice to see a lot of familiar faces. So today I'm going to talk about the Phenotype Differences Model. So it's a way to identify genetic effects with incomplete sibling data. And I just want to start off by acknowledging a key collaborator here, which is Clint Canopta. He's a PhD candidate at Stanford University. And then also, yeah, since arriving to University of Wisconsin, I've been working closely with Yushang and Q, going from left to right, Yushang and Q, Philip, and of course, Jason on this. So as an outline, like what are we going to talk about? First, I'm going to sort of raise this issue of kind of causation and correlation when it comes to genetic effects. So now we're back in this world where we're thinking about, DNA sequence. So not epigenome, not like RNA, but just sort of an allele change at a given genetic locus. So genetic causation versus correlation, and then sort of the existing tools, we'll move into the existing tools for making causal genetic inferences. And then I'm going to sort of propose a new method or a slightly augmented version of an old method and talk about sort of the assumptions that you have to make and sort of its efficiency, robustness, things like that. And then we'll conclude by talking about next steps. So as an introduction, Elliot talked about this, but GWAS have mapped genetic correlates of a wide range of complex traits. So ranging from education to height, depression, blood pressure, fertility. And the results are used to generate polygenic scores, PGS, or as Patrick would call it, a polygenic index, which aim to index individual genetic predisposition. So, I would argue that this sort of question remains, like, to what extent do, existing GWAS discoveries and then the subsequent PGS, built off of their weights, capture the causal effects of genes? And in particular, I think there's this threat of confounding from two sources. One is population structure or population stratification, and then the other is dynastic effects like genetic nurture. And if you want a paper that sort of outlines, the potential sources of phenotype-genotype association in human populations, I think this Young et al. Review in Science is a really great one. So quickly, I'll review sort of the issue of population structure. So an individual's genome contains granular information about their place within the structure of a population. Basically, where they fit in this big family tree of humanity. And this leads to potentially environmental confounding. So, allele frequency differences, the commonness of a certain genotype across subpopulations correlate with environmental differences. So, these environmental differences could be geographic, demographic, social. One thing I was going to flag about, with population stratification, I think we don't still fully know how big of a problem this is. And so hopefully some of these methods that I'm going to discuss will really sort of begin to elucidate it. And so, as you become to be familiar with the field, you'll just see this figure over and over again. But so here we have a bunch of genetic data collected in Europe, and this array of sort of million 0, 1s, and 2s at different spots in the genome. The first principal component is taken, the second principal component is taken, so just sort of a data reduction strategy. And then you plot them and you see, like, amazingly, it sort of recreates the geography of Europe. So this is sort of to highlight kind of like this really rich but also kind of cryptic information that exists within the genome, and it's not sort of clear how this might bias genotype-phenotype associations when we're interested in genetic effects. And in particular, so principal components like the ones used in the figure above, they've proven a pretty invaluable tool for controlling away population structure. In most analyses in the literature would control for between 5 and 20, sometimes many more. But there's an emerging literature showing that PCs really fail to fully eliminate the bias. And so I think, moving forward, we might need more new strategies to use in addition to PCs. I think it's actually maybe worth flagging. There's also some evidence that PCs might overcorrect a little bit, so that they might fail to fully eliminate the bias, but they also might be taking away, capturing some true genetic effects. So it's sort of an interesting problem there. So the other thing that I think is an issue here is dynastic effects. So a dynastic effect is a kind of an indirect effect. So an indirect genetic effect is the effect of one person's genome on another person's outcomes. So, for example, this was a paper published in PNAS. It's the effects of classmates' genes on my smoking or someone in the study smoking. But a dynastic effect is just a particular kind of indirect genetic effect. And indirect genetic effects, I think, have been most studied in sort of plant animal populations, less so in humans. And I think it's sort of an interesting open question in a lot of ways. Among family members, so genetic nurture is the effect of parents' genes on their kid mediated through the environment, not through genetic inheritance, is kind of the classic example. And there's this famous Kong paper that used a cool kind of identification strategy to highlight the probable existence of genetic nurture in a lot of traits. And so the issue is dynastic effects, like genetic nurture, they confound GWAS and polygenic score analyses due to the mechanical correlation of genes within families, right? So you're going to pick up some of the true direct genetic effect, and you're going to also pick up some of the sort of the dynastic effects. And so there's recent evidence really suggesting the existence of genetic nurture for many of the traits that social scientists are interested in. So educational development, socio-emotional development, mental health, things like physical health. So, I think we should probably, at least my prior is that genetic nurture exists for in a lot of cases. Here's a DAG that sort of gives the idea of genetic, of dynastic effects. So we have, grandparent, and then we have genetic inheritance by these dark lines. And then we have sort of the effect of each gene on some outcome of the child. And so this black line with the child's genes affecting their own outcome, that's sort of a direct genetic effect. That's often what we're trying to identify, I think, in GWAS. And then these red lines are examples of potential dynastic effects. This red line here from the parent to the child is a genetic nurture effect in particular. And so anyways, this is sort of like highlighting the problem because all of these genes are correlated. And so if we just associate this gene with this outcome, we're going to potentially pick up some of the red as well, as to the extent that the red exists. So from kind of this new information, we can kind of build this updated causal model. So we have some phenotypes, some outcome Y for person I and family J. And then we have an intercept term, and then this beta is the direct genetic effect. So it's the person I and family J's genotype. And then we added in a genetic nurture effect, but we can actually just kind of have this flexibly capture all dynastic effects as long as they're additive. And so, which is the sort of the parents in family J's genotype. And then we have the everything else basically term. We're calling it an environmental term, but we divided it into two levels. Basically, we have the family level environment, which is EJ here, and then we have an individual level environment. And it's important because I think we really believe a lot of the confounding happens at this EJ level. So in particular, like a key addition here is thinking about the dynastic genetic effects, and then also really taking seriously this like population stratification, this correlation between the family level environment and GIJ that's potentially going to confound just an ordinary least squares linear model. So how do we make causal genetic inferences? Existing GWAS and polygenic scores are likely confounded, maybe confounded. How do we identify the causal betas? How do we sort of test this? Well, what I propose is basically we use the same stuff as in non-genetic analyses, and others have proposed this too. So we should use our causal inference toolkit. Try to leverage only random genetic variation, which is by definition uncorrelated with population structure and dynastic effects. Unfortunately, with genes, we have kind of this like ultimate natural experiment, pun intended, right? Where conditional on his parents' genes, a child's genes are randomly assigned via recombination, and so really we just need statistical methods that allow us to condition on parental genotype. So in terms of existing methods, I think there's kind of two main streams. One is this family fixed effects approach. So we're taking the difference between siblings genotypes and difference between siblings phenotypes. So it differences out all family level variation in the outcome, which indirectly conditions on parental genotype, right? Because parental genotype doesn't vary within family, at least when there's sort of a classic nuclear biological family. So this requires two sibling pairs and two genotypes and two phenotypes, right? The other one is RDR, relatedness disequilibrium regression, or related methods to just condition explicitly on parental genotype. So this requires trios like mother, father, child pairs. So you need 3 genotypes and 1 phenotype. Or if you actually have phase data, you can do it with just 2 genotypes and 1 phenotype. And so these things work really well, but they have some limitations. And in particular, They both require dyadic genetic data within families, right? You need to have genotyped family members. And there's a dearth of this sort of data compared to at least sort of the ungenotyped, or compared to genotyped individuals. So for example, the UKB has 500,000 genotyped individuals, but only 16K are sibling pairs and only 10K are parent-child pairs. So like, you know, your power is going to be really diminished if you just sort of rely on the dyadic genetic pairs. And then also comparatively, you have less genetic variation within families. So, you know, we might need even bigger sample sizes if we're going to do like within family analyses. Plus, I think in the long run, we don't just want like a single well-powered unbiased GWAS for a given phenotype. You know, if these true causal effects are going to vary across place, time, you know, like across characteristics of a person, then sort of we're going to need, you know, to be able to run well-powered GWAS in a lot of even subsamples. So detecting heterogeneous effects is going to require even larger samples. So moving forward, how do we increase the sample sizes available for robust familial analyses? So I'm going to propose, or we're going to propose the phenotype differences method. And crucially, this requires only one sibling's genotype alongside two sibling phenotypes. So using phenotype difference, like when is this going to be useful? So I think that one place would be surveying individuals on the phenotypes of their siblings. So UKB, you know, if they had asked every person, don't only report your own education, but also report the education of a randomly selected sibling, or even better, all of your siblings, which would increase power. Then you could have turned basically, you know, what was 35,000 people that you could do for familial analyses into, you know, or 30,000, something like that, into all 500,000, right? Which would be a really big gain. Also, this phenotypic data already exists, so you can merge on phenotypic data of siblings from population registries, from health records, et cetera. So, you know, potentially you wouldn't even have to rely on self-report of sibling phenotypes. And also there's existing data sets that just have missing data, missing genetic data. People don't consent in the genotyping and they fall out of the sample. So the WLS, the Wisconsin Longitudinal Study housed at University of Wisconsin, has really great data on people, you know, sibling pairs from starting in childhood all the way to adulthood. But some of the people just stopped being impaneled later on, and they didn't survive basically, or they didn't stay in the study long enough to be genotyped. But if we have one of their siblings genotyped, now we can use the whole pair. So in explaining kind of how phenotype difference works, I think it's sort of important to outline what's going on with fixed effects, because the expression is very similar. So fixed effects, we have some outcome y, ij, and then we have basically a family level intercept. So this is, you know, some siblings outcome, they're the 1 or 2 sibling in family J. And then we just sort of estimate the association with this family level intercept between their genes and their outcome. There's an alternative expression of this, which you take the within transformation. So then we're just demeaning basically every unit within family, which is equivalent to having this family level intercept. And then, and actually in the case that we're talking about here, where there's only 2 units within each family, within each fixed effect, fixed effect is actually identical to first differences. So here we're just taking we're just subtracting y, like sort of the first person in each family, subtracting the second person in each family of their outcome. And then we're doing the same thing with the predictor, the genetic predictor here. So this is sort of fixed effects, right? We need 2 genotypes or 2 phenotypes and 2 genotypes. And we're going to see that phenotype differences is actually pretty similar to first differences, but it just requires a little bit less data. And we do that by relying on an assumption that probably is true in the case of genes, but not necessarily in the case of other sort of predictors. In particular, here's the general phenotype differences expression, which is we're still having the difference in the phenotypes on the left-hand side. We're adding in an intercept. This is actually important. And then we're going to basically, instead of having G1J minus G2J, so the sibling difference, we only have one sibling's genotype, but we're going to multiply it by the correlation of genotypes within families. And actually, this simplifies to something. This is looks kind of ugly right now, but it simplifies to something much prettier because we really know that this, in most cases, especially when there's not extreme sort of mating happening, that this is equal to 0.5. So really, we just get we can express phenotype differences as y1j minus y2j. With this intercept term, and then we're just multiplying, or we're just looking at the beta coefficient on G1J divided by 2. So this little transformation basically gets us back the answer that we want, but it only works with certain assumptions. So the first assumption is the same assumption that RDR uses and that sibling fixed effects use, which is just the random assignment of genes within families. So we have to believe that the covariance between genetic genes and then this within family environment is 0. So this assumption is not adding anything new. The next assumption is adding something new, which is that we have to believe that the standard deviations of the genotype sibling G1J is the same as the standard deviation of the ungenotyped sibling G2J. So this is really an assumption about selection into genotyping. So if we're actually just observing a random sibling of the two, then this is like sort of trivially true. But if you have really kind of nonlinear selection into which sibling gets observed in the sample, then it can cause problems. But we'll talk more about this later. And then finally, you have to know the correlation of genes within families. It's kind of like, I don't want to call this a genetic correlation, right? Because it's not like what we typically use the word genetic correlation for, but we need to know how correlated, two siblings genes are, which mechanically, in the absence of misordered mating, is equal to 0.5. I'm just going to blow through the derivation because that's a little in the weeds, and I'm the last presentation. It would be better to get out early. So comparative efficiency. So one thing that I'm saying is that this method will allow us to increase our sample sizes to make sort of causal genetic inferences. But like, it can't be, we're just sort of, you can just have less data and get away with the same result. That seems too good to be true. And you do take a power hit. So in particular, when genetic effects are small, so this is kind of the GWAS context, phenotype differences provides the same precision as fixed effects per genotype, though mechanically you have half as many genotypes per family. So you're going to get less power for each sort of family in your data, but you're probably going to be able to put many more families in your data, right? Because if we had surveyed, UKB people on their siblings' educational attainment, we'd have 500,000 rather than just, the 30-ish thousand that we have now. As genetic effects get larger, so this is more when we're like living in the polygenic score world, phenotype differences becomes comparatively less efficient to fix effects per genotype. But actually, in this case, it's probably not that big of a deal because we have a large genetic effect, so we're just pretty well powered to detect things anyways. So here's a figure that kind of shows this. Let's just focus on the left-hand side here. So on the x-axis, we have what we're calling phi. It's the r squared of the genetic effect within families. So this is different than the classic heritability, right? Because we're only using within family variation, and it, it just depends on how genetic effect, like how basically large the within family environment and the between family environment are compared to the genetic effects. But so when the genetic effects within family are very small, which we expect in GWAS for complex traits, right, where there's thousands of loci distributed all the way across the genome, each individual genetic effect is very small. Then the ratio of the standard errors of the fixed effects and the phenotype differences estimator is going to be equal to 0.5 for the same number of sibling pairs, right? So this is like, what if we took the same data set and then we ran fixed effects, and then we threw away one of the siblings' genotypes, and then we ran phenotype differences. And then as sort of the size of the genetic effects gets bigger, basically you do worse and worse compared to fixed effects, which sort of makes sense, right? Because you're not modeling, you're not properly modeling the genetic effect on the outcome. And so as the genetic effect gets bigger and bigger, then sort of not like modeling it accurately is going to basically cost you more in terms of efficiency. So, but the good news is here is like when you're at if the R squared of the genetic thing predictor, so in this case maybe a polygenic score is 0.5 within families, like you're probably very well powered to detect it even in a pretty small sample. So it's not a huge issue. So another way to think about it would be, well, this basically says the same thing. So we're just going to move on. So in terms of robustness, I think what things aren't a problem for phenotype differences that you might think are a problem. One is asymmetric classical measurement error. So just like, what if we survey people on the phenotypes of their siblings and they're just not as accurate as reporting in their own phenotype, which is likely the case. As long as this is measurement error, it's not an issue. If it's like sort of non-classical measurement error, we could get into some problems. There's also no problem with asymmetric bias. This is the work that Intercept is doing. So if respondents systematically over or underestimate their siblings' phenotype in terms of like, you think your siblings are taller or shorter than they actually are compared to self-report, that's actually not a problem either. Also, linear selection into genotyping is not a problem. So if genetic differences between individuals additively increase or decrease the likelihood of being genotyped, sort of versus the ungenotyped sibling, so making it into our sample, that's not a problem. But sort of different forms of selection that really distort the standard deviation of the genetic predictor, sort of the either genotype or polygenic score, that is more of an issue. And so just like quickly, kind of to finish things off, as a simulation, what we do is each one of these dots is a data set. We're simulating data using that causal model from earlier. And then we're fitting the 3 different models. We're fitting OLS, fixed effects, and phenotype differences. And what you can see, so here's the simulated beta. So we were hoping to like that the, in expectation recover the simulated beta. I mean, and what you can see is that OLS has bias. And this bias is a function of, we just sort of made it up, right? It's a function of like how much genetic nurture there is and how much population stratification there is. So this bias could be less than this. It could be even negative, right? They could be working in opposite directions. But the key thing is that both fixed effects and phenotype differences are recovering sort of the true genetic effects in expectation. But of course, you can see what we're doing here is we're using the same number of families to run these 2 models. And with phenotype differences, we're just doing it with less data, right? We're throwing away one of the siblings' genotypes. And that means that we take a precision hit, like I was saying. So you can see the dispersion of these sort of estimates is greater than the dispersion for the fixed effects, which is what we would expect. So then quickly, in terms of next steps, we've been applying this to GWAS with high blood pressure, and we've already sort of applied it in the PGS applications for 5 phenotypes. You can expand, or we're expanding the derivations and applications for G by E. You can use it for G by E in certain contexts, not all contexts, and then also Mendelian randomization. So you can, like this works for genetic IV, and then kind of creating bare bones R packages so people can apply this without having to code it up themselves. But anyways, I'm right at time, and that's all I have for you. But thanks so much for listening.
Did that generate questions from the audience that we'd like to talk about?
I have a question.
Yeah.
So that's super great, Sam. It's really interesting stuff. I think you're perhaps selling yourself short a little bit in the way that you've talked about it, in that, like in reality, you're not choosing whether or not to throw away individuals, right? And so I was wondering if you've looked at examples of data sets to which you might apply this, and what the power gains would be using the samples of genotype siblings versus what you could do if you had phenotype but not genotype siblings.
Totally, yeah. I mean, so and maybe the thought experiment there is like, well, what if there's 2 parts of it. There's like what do existing data sets currently have? What population registries currently exist? What does UKB currently have? And then there's like, well, what if we redid UKB, but we just happen to ask a slightly more questions? And like, so what if we, asked people about the siblings phenotypes in certain cases? But yeah, so I think you're right. Like, it's not that we're throwing away data, because of course you would never do that. But it's really that there's the data we're already using, which is sort of the fully genotyped sibling pairs. And then there's this other untapped data source of incompletely genotyped sibling pairs. And what Phenotype Differences does is it allows us to sort of tap into the second source. And even if you sort of the marginal gain of per sibling pair is smaller in the second source, yeah, overall it could be much larger. Or like, well, one, we're just gaining sort of data that we couldn't have otherwise used, but also, yeah, the much larger sample size means that probably you're going to have more precise estimates. To give a more concrete answer, so the project that we're working on, there's like very limited sibling phenotype data in some cases in different data sets, and I think we're able to have an effective sample size of what's equivalent to like 60,000 in a fixed effects case, rather than, so kind of twice that in a phenotype difference case. We're able to increase it, but maybe not as good as we would like to, given that, there's just not as much data on sibling phenotypes as we want.
Thanks.
Any other questions?
So I had a question. This is super cool. Sam, you went a little bit too quick through all those proofs for me to check your math, but otherwise I'm convinced. Here's my question. Do you, given what you're saying about the reality on the ground, we've got some data sets where we've got two siblings genotyped, and maybe in those same data sets we've got both phenotypes on siblings, some of which may have been genotyped, some of which haven't. And what we'd ideally like to do is use all the information together. So can you, so for instance, I guess one way, if I just thinking on the spot here, would be to make this a phenotype differences and genotype differences model and allow for missing data on the X's or the Y's? Or is it really as simple as just doing both analyses and then meta-analyzing them together at the end? What are your thoughts on kind of the way to capitalize on the maximum amount of information that's available in data sets that may have kind of different sources and different pieces?
It's a great question. So yeah, like, if you have sort of a mixed type of data where you can both do phenotype differences on some and then regular fixed effects on others, what do you do? Me and Patrick were actually talking about this recently with Alex Young, and Patrick was asking what kind of data sets, what kind of gains can you get? It's kind of funny, we're like hoping to work on something where we could potentially get a lot of gains, but the merging is a little tough, I think. I think the short answer is that there's a way to, well, when you have two genotype siblings, you would actually never now, I think, want to run just the regular fixed effects model. You would want to impute upward first using sort of the Mendelian imputation that Augie Kong and Alex Young have developed. And then you have a better powered, basically, fixed effects model, because now you have some data on the parents as well. And there's a way to basically bundle everything together in a single model, like the phenotype differences bit of it and sort of the sibling imputation. That's basically just like a mixed, like, linear model. So you can basically use the Alexander Young imputation framework as a way to rope both sort of fixed effects and phenotype differences together into the same model.
So you could have everything you want, Elliot.
And you can have your cake.
Yeah, I mean, pragmatically, that's what we want, right? Is to maximize the power given all available information from all sources. So that seems like pragmatically the thing that people would probably be most interested in. I mean, I guess I don't know. I'm not super familiar with the specific missing data patterns and sibling phenotypes and sibling genotypes. That seems like that would be pragmatically, the most useful to kind of incorporate them both.
Right. I mean, you can think with like something like iPsych or like a data set that it has like a lot of genotypes in a Northern European country. Then you can merge on the population registry for basically you have the full list of phenotypes for all of the sibling pairs. So then you have a lot of anyway, you have a lot of missing data basically.
Well, thanks everybody. I'm going to bring us home now that we're almost at the hour to just thank all our presenters and just kind of go back to the beginning and say, this was a conference on methods and big data, and I think you got a lot of that. So that was a success. And I also really enjoyed the fact that there was so much essentially open science here. There's a lot of discussion about R packages that were freely available and or under development, and a lot of data that is relatively easy to access for our larger community here on the call. So I think those are some good advances in this part of the literature. And finally, I'll just say, expect a survey about what you thought about our conference, I think early next week. And then later next week or in the following weeks, we'll send out a little reminder about the YouTube videos coming online and so on. So other than that, I really appreciate everyone's attention and presentations, and thanks to you all contributing to a successful conference.